Hello, everyone, and welcome to today's webinar, a spatially resolved transcriptional atlas of the murine dorsal pontine tegmentum. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by VizGen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Stefano Nardone, research scientist, Harvard Medical School, and Beth Israel Deaconess Medical Center for Life Sciences, and Corey Brannigan, Solutions Engineer at Heavy AI. Stefano, Corey, you may now begin your presentation.
Thanks for the kind introduction. And today I'm going to describe a project that is ongoing in Brad Lowell Lab. We are characterizing both spatially and transcriptionally the murine dorsal pontine tegmentum. But before starting, I would like to give an overview of where is the dorsal pontine tegmentum and which kind of a region it is. The pontine tegmentum is part of the pons, and the pons is a major division of the brainstem. As you can see, the pons is divided in two parts, a basilar pons that is more ventral, and the dorsal part that is the pontine tegmentum. And it spans from minus 4.9 millimeters from bregma to minus 5.8. And with spatial transcriptomics, particularly employing MERFISH, we characterize this region doing a section every 10 microns from minus 4.9 to minus 4.8 across, so from rostral to caudal. We have all sequential section in 4 mice. So just to give an overview of the project flowchart, we have a phase 1 in which we characterize transcriptionally this region, and we apply DRONG-seq, that is a single nuclear RNA-seq technology, droplet based, more or less like 10x. And after we cluster the cell and define the cell type, each cell type is defined by differential expression assigning a marker that is a unique gene that is expressed by that specific population. And then we use these marker genes in MERFISH to map this cell type in space. In phase 3, then, after, we partner with Heavy AI when we build a spatial viewer to visualize and query the dataset in real time instead of using a static plot. The dissection strategy we used for DRONG-seq was very diverse, and it basically point on two different regions. One is the lateral parabrachial nucleus that you can see in red, and another one is the Barrington nucleus that you can see in green. The two strategies were different because the Barrington nucleus has a high CRH expression, and so we use a reporter mice for GFP, and so we were able to pinpoint this region. And as you can see in the bottom on the right, we were able to microdissect under a fluorescent microscope. For the PBN strategy, we used an A14 mice that basically has like a stop cassette that flanked by 2 LOXP sites. And only when you inject an adenovirus that has CRE in the NTS, basically it happens the recombination, and the 3D tomato, that is the protein fluorescent you can see, is expressed. Fibers from the NTS, they project heavily to the lateral parabrachial, allowing us to dissect and to have a guide during the dissection. So after we did DRONG-seq, we clustered all our cells. As you can see here in the T-SNE in figure C, we were able to characterize neurons, choroid plexus cells, vascular leptomeningeal cells, two kinds of cells, these are type 1 and type 2, the microglia and perivascular macrophages, epiendemocyte, and so on. And of interest, neurons accounted for 60% of the entire population, and we provide around 150,000 nuclei that pass the quality control. Next step was to doing the neuronal clustering of the dorsal PNTG. We are interested in the neurons, so we separate only the neurons. We isolate the neurons from the glia and the non-neuronal cells, and we divide them in excitatory and inhibitory. The excitatory neurons, basically they express BGAT1, 2, and 3, and plus we included in that also chat neurons, cholinergic, serotoninergic neurons, and noradrenergic neurons, while in the inhibitory neurons we have like VGAT and as marker, but also they express some transporter for the glycine, so some of them are also glycinergic, and we have a few clusters of hybrid neurons. That means these neurons, they express both excitatory and inhibitory transporter, and therefore their outcome, electrophysiologically speaking, we don't know which is. On the bottom you can see on the y-axis the genes, while on the x-axis the clusters. So we identified a total of 33 and 29 clusters, both for the excitatory and inhibitory neurons. However, the brain and single cell technology is a very powerful technology to characterize the transcriptome of the brain, but it doesn't supply what is like a spatial resolution. So we have like a sort of information from each population, but we don't know where it's located. And here comes the spatial transcriptomics, and particularly MEFISH. We partnered with VIXGen, and we apply MEFISH to this region. The most important things to understand is that we choose the highly variable genes. It means genes that are markers that are expressed only in certain specific population. And then we use a panel of 315 genes to locate these populations. Here you can see a MEFISH tissue cutting of 10 sequential sections from rostral to caudal. After we cut this 10 micron unfixed sections, we go through all a process that's been done by VIXGen in their lab, in which there is like fixation and permeabilization, cell boundary staining, hybridization, then gel embedding, and digestion and clearing. After this, the output is basically a list of detected transcript, mosaic images in TIFF, in which you can see the data, transcript per cell matrix, and metadata and cell boundaries. So these are the cell boundaries, so-called segmentation, and we define the cell boundary of the dorsal pontine tegmentum in the bottom right, and so we pull only the cells and the transcript inside that region, because we were interested only in that region, and we choose the gene to be hypervariable for that specific region. After that, we did more or less the same approach we use for single cell sequencing, and we cluster all the cells. We had only in this region nearly 700,000 cells that passed the quality control, but on all the slides we have 6 million cells in all the 49 slides that we imaged. Then we divide the neurons in excitatory and inhibitory, as we did before, and what we did is like finding the unique marker for each cluster, as we did also in single cell RNA-seq. One of the major negative side of clustering all neurons of a big region is that we lose some granularity in the clustering. So to gain more granularity and to build an atlas that is of interest of investigators, that they go and they perform stereotactic injection in specific regions, subregions actually, we then divide the dorsal pontine tegmentum in 4 subregions that we anatomically defined. Another approach we applied is to perform a canonical correlation analysis between Merfish and Dongsec clusters in the dorsal pontine tegmentum. This is very important because when there is a direct correspondence, as in some of the cases, you can infer genes that you didn't assayed or probed with Merfish from the Dongsec dataset. So we found that 71 out of 85 Merfish identified clusters correspond to 49 to 59 Dongsec identified clusters in the mouse dorsal pontine tegmentum. So as I said, we divide this region in 4 atlas. The first is the colliger fuse, second is lateral parabrachial and medial parabrachial. Then we have the mesencephalic trigenimal nerve, the prelocus ceruleus, locus ceruleus, the Barrington nucleus. And in the 4th atlas is more the medial region with the lateral dorsal tegmental nucleus, the ventral tegmental nucleus, the dorsal tegmental nucleus, central part, pericentral, and the posterior part, and the sphenoid, nucleus incertus, and so on. Today we're going to talk about the Atlas 2, just to give an example of how we build the parabrachial complex atlas. First of all, the parabrachial nucleus complex is formed by the colliger fuse and the parabrachial, and is an important relay in this region for all the input that comes from, for instance, stretch or nutrient sensing from the gut, through the vagus nerve, the NTS, and then the parabrachial, or through the spinoparabrachial tract. They can sense warm, cold, pain. And then after, also energy status through the arcuate, then the PBH-MC4 receptor through the PBN to an unknown population. After the PBN project to high order sites, where they set the emotional balance to motivate the behavior. So in this slide, what we did is basically defining, as we showed before, some boundaries on the DAPI, and then we pull only the cell and the transcript that were in those boundaries. After we clustered all the cells, around 80,000 cells, and we plot as Voronoi plot, a sort of cartographic map, all the clusters that are color-coded, as you can see on this name on the right. We divided the cluster in rostral, caudal, and scattered according to their frequency. As you can see on the bottom, you can see the frequency of the clusters, and you have a Bregma level on the x-axis and the cell count in percentage on the y-axis. For instance, if you wanted to pinpoint where is protodynorphin, that are clearly these neurons in orange here, but you're not sure where are on the cartographic map, on the Voronoi plot, you may see that here there is a bump. It means there are more neurons here in this part, and so it's a minus 5.35. That is this level. The same is for calcaneurons, that we know the well-known population in the rostral parabrachial nucleus, and then they go ending toward the caudal part and mostly shifting in the medial part. So this helps a lot to visualize what our results and to get insight. However, this static plot, especially for population that are very rare and represent less than 1% maybe of the dataset, are not very indicative. Here in this other slides, we then characterize the top 2 markers for each of the clusters. And then we ask that question, if mice and human neuronal subtype reveal a high degree of transcriptional conservation in this area. We retrieved from Siletti et al. From Linarson lab, single nuclei data from human from the same region. And as before, we clustered them. And as you can see, the percentage is 66% of the neurons. That is very close to the one we had before pointed out with Merfish. We plot the same genes, and we divided it excitatory and inhibitory, defining genes that are marker genes and the number of clusters for each group. After we asked the same question before, and we calculate the correspondence between cluster for single nuclear RNA-seq in human, PTNG, versus drunk RNA-seq in the same area. And we found that there is a very high correspondence. You may see here, for instance, Calca, that is one of CGRP, one of the main and most studied cluster population in the parabrachial nucleus, correspond to a Calca population in the mouse. So we went actually back in time, and we looked at if really there is this Calca CGRP neurons in human, and we found a paper in which actually they identify these neurons. They are in the same position where we found in the mouse. It means in the external lateral part of the parabrachial nucleus. And so after we looked at their transcriptional profile of the top expressed genes, and we found that their transcriptional profile is quite similar, if not identical. Here on the top, on the dot plot, you may see PBN, snRNA. These are all the nuclei of the parabrachial nucleus in human, except Calca cells. And these are all the Calca cells. It means the Calca cluster number 2, this one, you can see here, without any other cells for any other cluster. And the same has been done in drunk, where you see Calca in mouse here, and you can see as the same pattern of expression, and also in Merfish. Where there is a white spot is because the gene was not detected in Merfish because we didn't test it. For my part, I wanted to thank you, my boss, Beth Lowell, and all the co-authors of this work. And I will give the floor to Cory. Thanks a lot.
All right. Yeah, thank you a lot, Stefano. Just for context, Stefano and I have been working together on his work within heavy AI and his research. So I've been supporting that. So yeah, before we get into the tool itself, I was just planning to give a little bit of an overview of what we do here and how we've been helping out with Stefano's research. So to begin, heavy AI is a technology that allows organizations and users to interactively analyze and visualize their largest data sets. So we're talking about scales of 10 million to tens of billions records, delivering results instantaneously. So I don't need to tell anyone that data volumes have exploded at a remarkable rate. And those volumes are now outpacing gains in traditional computing power. So having enough data to educate our decisions is no longer the problem. The problem has shifted to analyzing and making sense of that data. So Heavy AI was founded in 2013 when our founder, Todd Mostak, was in a graduate program study, actually at Harvard as well. He was looking at the Arab Spring through the lens of Twitter. So he quickly felt limited by the SQL tools that was available to him. He would typically run an analytical query overnight and either find it still going in the morning, failed, or require some change, which would start the process over again. So we built a querying engine that was powered by GPUs, taking advantage of the parallel processing they could do with data in memory. So we have funding from top-tier venture capital firms and have many customers and partners in the Fortune 500. For example, we enhanced S&P Global's Energy Studio for understanding oil and gas investments. We helped Charter with offloading mobile devices to hotspots and other antennas. And we worked with SoCal Edison, which is one of the largest utilities in the country, to mitigate wildfire risk around their power lines. So we solved these challenges by allowing customers to work at scale. We believe that people make the best decisions by literally seeing all of their data together in one place, even if it comes from multiple sources and is continuously changing. So like I said, extremely large data sets in the orders of hundreds of millions to tens of billions of records are no stranger to us at all. So we want you to bring your data as it is. That means no downsampling, no pre-aggregation or data reshaping required. We can import data sets directly into heavy AI or connect to them where they currently live in the infrastructure that you've already invested in. We can organize it into dashboards and charts, allowing you to interactively find hidden opportunities and risks. You can find patterns and anomalies, understand your behavior over time, compare scenarios between different models or data sets, and identify and measure relationships between them. Typical operations you want your data analysts to be able to perform. The difference here is that these results are delivered instantaneously, enabled by rethinking the analytics solution to leverage the power of GPUs. Heavy AI enables you to speed up your SQL queries over 100x and allows you to visually interact with your data. When you compare versus the cost of running these jobs in a traditional CPU-based environment, it's up to 10 times cheaper and can be 10 to 100 times faster. So the heavy AI stack includes both an end user product and a platform accelerator. And we leverage that GPU power throughout the stack. We can connect or import data directly from data warehouses, data lakes, S3, Parquet files, and through standard ODBC connections. You can use the full stack or utilize different parts to power third-party applications, such as interfacing with machine learning workflows, accelerating other BI tools, or building custom rendering applications. So we have 4 basic components. First, we have HeavyDB, and that's our SQL engine that's tailor-made for GPUs. That powers HeavyRender, our rendering engine for delivering visuals of large amounts of data. And on top, our interactive no-code visual environment, HeavyImmerse. So this is a dashboard building environment that allows engagement by more non-technical users and can be shared out more broadly. And we also have HeavyConnect, and that's this is our foreign storage interface, and that allows you to connect to a data source, leave it where it is. It reads the data directly into the GPU memory cache. So just for reference, here's a number of customers whose decisions we are supporting today, along with a handful of technology partners that we work with. As you can see, a lot of large enterprises and also plenty of work on the intelligence and federal side trust us with their data, with doing very critical analysis. So getting into the interface itself, here's just a quick preview of what Stefano will show. While the main chart on the left-hand side is typically used for geospatial data, anything that uses point coordinates are supported. And so as you can see when zooming in, we have both points and polygons in this view. And so now I'll hand it off to Stefano to show how this works interactively.
Thank you, Cory. So let me share the screen. Just 1 second. So I guess everyone can see now the dashboard. So this is an interactive dashboard that we designed with a heavy eye, and that is holding inside the data that we just talked in the presentation. As you can see, we plotted the segmentation just for our dorsal pontine tegmentum, and also the statistics cell by Segret clusters, I mean the cellular statistics, are and regards only the dorsal pontine tegmentum. However, we plotted all the transcript, and in the future we will plot even all the segmentation. And we were able to see, for instance, like in our dorsal pontine tegmentum segmentation, around 13,000 cells only on this slide. And on the overall section, we have a total of more than 65 million spots. So let's navigate inside to see how does it work. As you can see, it's all interactive. I mean, the statistic changes when you zoom in or zoom out. And when you do it, you may, at a certain level of zoom, you may start seeing actually the cellular structure. So it works basically that it's possible to select structure from here. For instance, we wanted to know only this population, and we can get rid of the genes, for instance. So this is the dorsal raphe, and here there is the prediction in DRONC. It means that basically in between the two technology there is agreement on that in the same cluster in marefish has been characterized by DRONC. So basically, if we didn't characterize any gene in marefish, we can go back to DRONC and to single nuclear RNA-seq means, and check that gene, because we know that there is a correspondence done by CCA from the two populations. So here you can see both the dorsal raphe that is marked by TPH2 and its transporter of serotonin, and for instance you can see also some other population. This should be the DTN. You may see the ependymocyte. But then what if we want to overlap the gene on this? So we can go in custom selection, and we can select, for instance, some of the gene we wanted to see. Just I remember some marker of the ependymocyte. You will have your marker plotted here. Now you cannot see them because it is covered. You see how this population is basically highly enriched for these two markers. Of course, then you can have a broader view of the neuronal organization just plotting all the cells in this region, and mostly actually taking out the genes. So as you can see here, each cluster has its own spatial pattern. And correspond basically to, this is the VTN, and correspond to a specific region. So there is like a spatial organization in this region. So if we want to go and look at one of the 4 atlas we did, we take a look at the parabrachial nucleus. In the parabrachial nucleus, you may see this is quite caudal part of the parabrachial. Precise is minus 5.35 from Bregma. And that's before we can take out the genes. In gray, you can see the non-neuronal cells of the glia. We were a little bit abundant with the boundaries. But one very interesting thing is this function of lasso that we have. Because if you are interested, for instance, in this cluster here in the T-snare, but where you don't know where it's located, you can do mostly a lasso on the my mistake. You can do a lasso on the specific cluster, and you will see only this cluster. And this can be done for many other clusters, but most important, you can see that there is also here an organization of all these clusters. Some are like more dorsal, some other are more lateral, and any cluster is characterized by a specific marker. Okay. So if you want to change slice, we are interested in going, for instance, to another atlas. This is possible. And you just need to change here the atlas name. Let's see Atlas 4, for instance. Now, all the cluster and the statistica refers to this more medial part of the brain. In which we actually can see a very nice organization of the dorsal tegmental nucleus. In here. This is the ventral tegmental nucleus, and this is the dorsal. And also here you may see a very particular, you see, spatial organization. So after this, I mean, we can start taking some questions. I will stop sharing the viewer.
All right. Well, thank you, Stefano and Corey, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question asks, were there other AI applications you considered before selecting heavy AI?
Okay, so this is a question for me, I guess. So basically, yes, we tried other applications, but actually with the heavy AI, we found that the rendering and actually the speed and the number of data that we can plot are far higher than other company. So this is why we partner with them to create, because data in the future is just supposed to grow. And so even if their capability now exceed what are our data that we are inputting in their dashboard, in the future we will have 1 billion of data that need to be visualized. And so I think AI is a very strong partner for this.
Great, thank you. Next question here is a two-part question, and this asks, How many genes were in the MERFISH panel, and did you get help selecting the panel?
Yeah, sure. In the MERFISH panel, there are 315 genes. As I said, we selected the genes, the hypervariable genes. It means genes that have a high variability among all the cells. And these usually are those that by differential expression then we identify as markers, means genes that are expressed only in a specific cell population, that is glia, non-neuronal, or neurons. Yes, we did get help from Vizgen in selecting the panel, and especially because we matched our gene with the most expressed isoform in the brain. And so because we do this, we are sure to have like a reliable results, because some genes they have different isoform, some isoform may be non-expressed in the brain, and so this is why we have like an RNA-seq dataset that allows us to understand which is the highest isoform for each gene we wanted to select.
Great. Next question here asks, besides data analysis and visualization of MERFISH data, do you see other uses for AI in biological research?
Corey, do you want to try to answer this?
If you want to take it, I'm not the
okay, so
bio expert.
So yes, there are other uses for artificial intelligence in healthcare. For instance, like in drug discovery or in pathology, there is like a lot of application for artificial intelligence. Yes, for sure.
Great. Next question, we have another two-part question here. Is this running on a desktop, and how is the software deployed?
Yeah, that one I can take. So this is software is deployed on premises, and so generally that'll be on a local server, or you can use any of the different cloud providers and spin up a machine on there. So it'll require just anything with a NVIDIA GPU that's just spec'd to the amount of data that you'll be working with. So yeah, everything can live within your cloud or within your on-site hardware. So no worries around data security there.
Great, thank you. And another question for you, Corey. What preparation was done for the data that is being displayed before importing into the platform?
So very little. We received this data from Vizgen, and I think just a couple adjustments that were made were just around the point data of just kind of scaling the values a little bit, so they were more manageable to work with on our mapping chart. And other than that, we were able to just import directly into Heavy AI and immediately perform the visuals from there.
Awesome. All right. Well, we are going to go ahead and wrap up there. So I want to thank you again, Stefano and Corey, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Vizgen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you had provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.